Now let's get back to the Wikipedia page on biology, and this time look at macromolecules. And further information are on the macromolecule or biochemistry wiki articles. So let's go over here. Yeah, so molecules such as sugars, amino acids, and nucleotides can act as single repeating units called monomers to form chain-like molecules called polymers via a chemical process called condensation. For example, amino acids can form polypeptides, whereas nucleotides can form strands of deoxyribonucleic acid, DNA, or ribonucleic acid, RNA. Polymers make up 3 of the 4 macromolecules, or very large molecules, which are polysaccharides, lipids, proteins, and nucleic acids that are found in all living organisms. And here's just a table of the major macromolecules. So here's the macromolecule, the polymer, proteins, and its building block or monomer is amino acids, and bonds that join them, they're called peptide bonds. And then here is nucleic acid. Yeah, these are DNA and RNA. But yeah, so the bonds that join them are phosphodiester bonds. And so DNA is a type of nucleic acid, and this is comprised of the monomers nucleotides, which is a phosphate, ribose, and a base. And a base, which is adenine, guanine. Adenine, guanine, thymine, and cytosine. And RNA has nucleotides as well as monomers, and they involve a phosphate, a ribose, and this one is RNA is ribose, and then DNA is deoxyribose. So this should be a deoxyribose. Yeah, so deoxyribose. So this is a bit off there. Yeah, so I'll just put that actually in here. I'll just erase this and put it right here, the major macromolecules, and then just put it like this. Yeah, just type here, DNA should have deoxyribose, not ribose. But nonetheless, you could consider them both as riboses. Yeah, just be more general, but yeah, but more specifically, it's deoxyribose, so it's a ribose with a oxygen removed. So then RNA is the phosphate, ribose, and a base, adenine, guanine, and then uracil, and cytosine. So the only difference is, the difference is thymine and uracil, and also the ribose. This is deoxyribose, and this is, DNA should have deoxyribose, not ribose, and then RNA has ribose. And then going further, so polysaccharides, or just carbohydrates. So the building block is the monomer is a monosaccharide, and the bonds that join them are glycosidic bonds. And then lipids, they're unlike the other macromolecules. Lipids are not defined by chemical structure. Lipids are any organic nonpolar molecule, or a combination of them. And so nonpolar like oil and so on. So some lipids are held together by ester bonds. Some are huge aggregates of small molecules held together by hydrophobic interactions. So yes, so lipids are more aggregate based, as opposed to these ones are all chains of small specific monomers, or types of monomers. So going further, so each macromolecule plays a specialized role within any given cell. Some polysaccharides, for instance, can function as storage material that can be hydrolyzed, or which involves chemical bonds broken by water to provide cells with sugar. Yeah, so basically carbohydrates break, the bonds break and provide cells with sugar, so break in water and so on. Lipids are the only class of macromolecules that are not made up of polymers, and the most biologically important lipids are fats, phospholipids, and steroids. And just note, phospholipids, also known as phosphatides, or phosphatides, I think that's how you pronounce it, are a class of lipids whose molecule has a hydrophilic, or attracted to water, a head containing a phosphate group, and 2 hydrophobic, which are not attracted to water, tails derived from fatty acids, joined by a glycerol, which is a multiple OH hydroxyl molecule. Yeah, so it's basically has a hydrophilic, it likes water, and then tails that don't like water, 2 tails. So it looks like this. So there's a 2 tails right here, hydrophobic tails. like that, and this is a phospholipid, and they form a lot of the membranes and so on. So here is, over here, hydrophobic head, here a hydrophilic head, which likes water, and hydrophobic tails that don't like water. So if this is water on the outsides and inside, these ones would go to the water, and these tails would move away from the water, so you can form a membrane. So extracellular space outside here, and then inside the cell. This is outside of the cells, this is inside the cells, a lumen of cells, just opening inside. So phospholipid arrangement in cell membranes. Lumen is the inside space of a tubular structure, such as an artery or intestine or even a cell right here. A steroid is a biologically active organic compound and a type of lipid with 4 rings arranged in a specific molecular configuration. Steroids have 2 principal biological functions as important components of cell membranes, which alter membrane fluidity, and as signaling molecules. And there is over here. Yeah, so you could see the 4 rings right there. So A, B, C, D. So there's 4 rings. So structure of Cholestane. Yeah, I think it's pronounced Cholestane. Yeah, so and it's a steroid with 27 carbon atoms, and there's all 27 there. Its core ring system A, B, C, D is composed of 17 carbon atoms. So it has a specific molecular configuration, and this has the 4 rings over there. That's fascinating stuff. And membrane fluidity refers to the viscosity of the lipid bilayer of a cell membrane. Yeah, the viscosity or thickness, or yeah, just the strength or how sticky it is, etc. of the lipid bilayer, and that's what this steroids helps with, as well as signaling molecules. And the lipid bilayer or phospholipid bilayer is a thin polar membrane, positive charge on one side and negative on the other, made of 2 layers of lipid molecules, and that's pretty much it. There is a lipid bilayer over there, so there's 2 layers. Like that, so the bi is the 2. And here is some setups of this. Yeah, and here is the 3 main structures phospholipids form in solution. The liposome, which is a closed bilayer. So you have a closed one like this. So there's the bilayer. So there's a layer here and layer around there. So then you have an inside gap in between. So that's a closed bilayer. And then the next one is the micelle. So this one right here, the micelle or micelle right here. So there is no extra no extra phospholipids inside. It's just the tails are inside, so that looks like that. Then you also have the bilayer sheet right here, which has 2 layers like that, and you can have a long sheet like that. So that's fascinating stuff. And yeah, these come up all throughout this video and throughout biology. So going further, so proteins are the most diverse of the macromolecules, which include enzymes, and then transport proteins, large signaling molecules, antibodies, and structural proteins. Finally, nucleic acids store, transmit, and express hereditary information. And here's MES note on antibodies. So right here is the proteins are the most diverse, which include antibodies. So an antibody, or also abbreviated AB, also known as immunoglobulin Ig, is a large Y-shaped protein used by the immune system to identify and neutralize foreign objects such as pathogenic bacteria and viruses. The antibody recognizes a unique molecule of the pathogen, which is the organism that can produce disease, called an antigen, and this is abbreviated AG, or antibody generator. Going further, so to allow the immune system to recognize millions of different antigens, the antigen binding sites at both tips of the antibody come in an equally wide variety. In contrast, the remainder of the antibody is relatively constant. It only occurs in a few variants which define the antibody's class or isotype. And you have IgA, IgD, IgE, IgG, or IgM. So the types of antibodies over there, or the class of it. So here is the basic theory or the structure of this antibody here. So let's make it a bit smaller to fit in. So each antibody binds to a specific antigen, an interaction similar to a lock and key. So if you have this antibody like this, and it has a specific antigen binding site, and these are the antigens, and it can connect to it like that. Then this would connect with this antigen and neutralize it, and so on. Or allow the body to respond however it can respond to. And the primary goal is to identify and neutralize foreign objects such as these pathogens. Going further, right here. And yeah, now let's look at the structure of proteins, or just this is a hemoglobulin protein. So the primary yeah, these are the different structures of the protein right here. So here's a primary structure right here. So the A, primary, B, secondary, and C, tertiary. Actually, I think it's pronounced tertiary, and then D is quaternary. Quaternary. Let's try that again. Quaternary. I have to Google pronounce again. Structures of a hemoglobin protein, or I think it's pronounced hemoglobin protein. Yeah, where hemoglobin, or HB or HGB, is the iron-containing oxygen transport metalloprotein, which contains a metal ion cofactor in the red blood cells, also called erythrocytes. So yes, we'll get to that in a bit. So here's the different structures. Here's primary structure, just a chain of amino acids. Then here is the secondary structure, which is a pleated sheet. A plated sheet, or just folded sheet. So you have it. Here's an alpha helix, a type of helix. So like that, there's a chain. There's a chain of amino acids. Then you make it into a sheet like this, or just initially go to a helix like this. And then you have a folded setup. So this is folded in 3D. So you could have folded like that, or pleated, which is folded sheet like that. And these are bonded on the opposite ends like that. So on these ends there's bonds, and so on. And then here's a tertiary structure. Yeah, and this tertiary structure has just loops all around, and these heme, or hemi. Actually, I think it's pronounced heme. Yeah, so these heme units, I just put it here, just over here for reference. So heme equals precursor to hemoglobin. Yeah, so hemoglobin or hemoglobin. I think it's hemoglobin is the proper pronunciation. And yeah, then now you get a whole bunch of these together. So then you have the quaternary structure. This is a tertiary structure. So you have this folded. The next one is a loop it all around, and the next one is you have many units of these loops around like this, and you have a quaternary structure. And then you have this hemoglobin globular protein, globular or spherical. I'll just put that like this. Globular equals spherical, or ball, etc. Ball, globe, etc. So yeah, you have this a giant blob like that, or globe. And yeah, going further, yeah, so the hemoglobin is iron-containing oxygen transport metalloprotein, contains a metal ionic cofactor in the red blood cells, also called erythrocytes. So red blood cells, RBCs, also referred to as red cells, red blood corpuscles, or I think it's pronounced corpuscles, in humans or other animals not having nucleus in red blood cells, and also hematides, erythroid cells, or erythrocytes, from the Greek erythros for red and kaitos for hollow vessel, with cyte translated as cell in modern usage, are the most common type of blood cell, and the vertebrates, which are a class of animals with backbones, principal means of delivering oxygen, O2, to the body tissues via blood flow through the circulatory system. Yeah, so these structures right here, so just to go in depth more on them, so the biomolecular structure is an intricate, folded, three-dimensional shape that is formed by a molecule of protein, DNA, or RNA, and that is important to its functions, different shapes of different functions. And going further, the structure of these molecules may be considered at any of several different scales, ranging from the level of individual atoms to the relationships among entire protein subunits, which are just individual protein molecules that assemble with other protein molecules to form a protein complex. So the subunits of a bigger protein complex, or aggregate. So a protein complex or multi-protein complex is a group of two or more associated polypeptide chains. Yeah, so yeah, two or more associated polypeptide chains, which are just proteins. Protein complexes are a form of quaternary structure. So you have a whole bunch of these tertiary structures, and you make a whole bunch of these as a quaternary structure of these polypeptide chains. Yeah, so these are all just polypeptide chains right here. Primary structure, chain of amino acids. You have a whole bunch of those, you're going to get some long protein complexes. Yeah, so this useful distinction among scales is often expressed as a decomposition of molecular structure into four levels: primary, secondary, tertiary, and quaternary. The scaffold or support structure for this multi-scale organization of the molecule arises at the secondary level, where the fundamental structural elements are the molecule's various hydrogen bonds. So these ones here, there's a chain, and then these are all bonded by the hydrogen that forms the basic structure of these, of these organization of the molecules all, and that's at the secondary level. So there's a fundamental structure set up. Yeah, so now let's look more at these types of structures. So over here, over here we have primary structure. This is for a protein structure, and the other one is nucleic acid, just to get the overview. So here's primary structure, just the chain of amino acids linked like that. And then you have secondary, you have a shape like this, and it's connected by hydrogen bonds. Then tertiary, you get a whole bunch of these, and then you have different types of secondary structures combined together. Then you have a quaternary, you have these all bunch. So you have this all connected, more of these chains all together. And you have this monomer 1, monomer 2, monomer 3. So all different types of monomers connected into one giant structure like that. So this interactive diagram of protein substructure using this software, etc. And there's interactive for nucleic acid structure, primary, secondary, tertiary, and quaternary using DNA helices or helixes, and examples from this right here, these types of molecules. So anyways, going over here, so you have the primary going from the 5N to the 3N. These are the nucleotides, or the nucleotides with the base pairs over there, GC, GU, over all the way to there. Yeah, this one has a U, so that's uracil, so that's going to be an RNA strand. And you have secondary, you have different types of connections. So you have a helix, you have a stem loop like that, and then a pseudo knot. It looks like that, so it's not like that. And then you have the tertiary, where you connect a whole bunch of these, and you have these types of connections like that. Pseudo knot, stem loop like that, and very interesting type setup. And then you have the quaternary setup, where you have a whole bunch of these all grouped together. And this is for DNA, and that's a histone protein. We'll get to that in a bit. And then here is the RNA. We have different monomers groups, so monomer 1, monomer 2, etc. And so this histone protein, this DNA is wrapped around it. So histones are proteins that act as spools around which DNA winds to create structural units called nucleosomes. Yeah, so it's like winding a string or a thread. So that's the spool. So that's as a spool, which is very fascinating stuff.